Welcome, Brookings APBIO students, to the last podcast in our series on macromolecules, and we talked about carbs, proteins, and lipids, and this is our last group, nucleic acids. If you think about those other ones, that we talk about them as food groups, like how many carbs did I have, or I'm on a low-carb diet, or did I get enough protein today? And we talked about fat in our food, but we don't really usually talk about nucleic acids as a food group. The reason is that, remember, nucleic acids, the Na, is part of a DNA molecule. That's what DNA is, a nucleic acid. And so if you're eating something that was once alive, all living things have DNA in them. So we don't really worry about not getting enough DNA or nucleic acids in our food. So we don't really talk about that as a food group. But it is an important molecule in living things, obviously. And so we're going to talk about this group and what it's made out of and what we use it for. The main functions of nucleotides, nucleic acids, are information storage, making proteins, and as energy storage and transfer molecules in cells. And so if you look, the little building block that I use to make DNA and RNA with, nucleic acids with, are called nucleotides. And it's a monomer subunit that we're going to use to make a bigger polymer. Nucleic acids are polymers, just like protein is a polymer of amino acids. Nucleotides are the building blocks we use to make nucleic acids. The nucleotide subunit looks like this. It has a sugar component, has a nitrogenous base component, and it has a phosphate group. And by changing what I put in for my sugar component and what I use as my nitrogen base, I can make different subunits, different building blocks that are used to join together to make a nucleic acid. If I look at this little part of the molecule, the sugar and phosphate alone is called a nucleoside with an S. All three of those, I'm sorry about the computer jumping, all three of those together make it a nucleotide subunit. It has a nitrogen base, a sugar, and a phosphate. If you look at the choices for sugars I have, depending on what kind of nucleic acid I want to build, I can put deoxyribose sugar or ribose sugar into that sugar spot. Notice the difference between these two. This oxygen is missing. It's deoxy. I've lost an oxygen off of ribose to make deoxyribose. The nitrogenous base choices I have for making DNA or RNA are the purines, two carbon ring shaped molecules, the purines, adenine and guanine, or pyrimidines. Pyrimidines have a one ring structure. Cytosine, thymine, and uracil are my pyrimidines, my choices for nitrogen bases. Notice the difference. Remember, A's and G's and C's and T's are used to make DNA. A's and G's and C's and U's are used to make RNA. And look how closely the T's and the U's are to each other structurally. The difference is that one methyl group there on the ring. So purines, double ring nitrogenous bases. Pyrimidines are single ring nitrogenous bases, and they're important in seeing how this molecule goes together. We're going to take now, and here's my one nucleotide, and here's my next nucleotide, and they're joined together with directionality. If you look at the way the components of the nucleotide are attached, the nitrogen base part hooks onto the number one carbon in our sugar ring. And remember when we talked about sugars, we number the carbons in the ring starting with number one next to that aldehyde oxygen. That's our number. So carbon number one, number two, number three, number four. This is a 5-carbon sugar. And so nitrogen base is attached at the carbon 1 spot. The phosphate group is attached on the number 5 carbon. Remember, that kind of sticks up above the ring like a flag. And now my phosphate is going to attach to the next component by what we call a phosphodiester linkage. It's a bond that forms between the phosphate in this molecule and the OH group here. And we're doing a dehydration synthesis, just like we did before. Pull out a water molecule, make a bond. Notice now each strand has directionality. We're adding this little subunit onto the end of the completed chain, and we add on we're always adding onto this number 3 carbon. And so if I look at how the chain runs, it has a 3', we call it a 3' end, and a 5' end. The closest carbon in the backbone here, now notice phosphate, sugar, this is going to make the backbone of our molecule. The nitrogen bases stick out to the sides. If I look at the backbone, my number 3 carbon is facing this way, and my number 5 carbon is closest to this end. So we call it, this is my 5' end, this is my 3' end. There's directionality to the strand. So here's my little subunit, and I'm going to make a strand by joining those pieces together that alternates phosphate, sugar, phosphate, sugar, phosphate, sugar. That makes the backbone, the sides of my DNA ladder when I make DNA. And then the nitrogen bases stick out toward the middle. Okay? And if I'm going to make RNA, I use ribose sugar. If I'm going to make DNA, deoxyribose goes in for my sugar spot. And then nitrogen bases are my choices for making, in this spot here, for making, depending on which, if I'm making DNA or RNA. So DNA, that's our gene code molecule. It's the carrier for our genetic information. Majority, 99% of living organisms have DNA as their genetic code molecule. Deoxyribonucleic acid. It's got a deoxyribose sugar. My nitrogen bases are A, C, G, or T choices. And it's going to actually make two-stranded. It's a double-stranded, we call it a helix. There's a strand that runs down one side, and there's a strand, and they come together in the middle. Notice the nitrogen bases are what make the rungs of the ladder. The phosphates and the sugars are making up the backbone, the sides of the ladder. Let's go back one. If you remember from bio, that connection across the middle is a hydrogen bond that holds the two sides together. There's hydrogen bonding between these two nitrogen bases. And remember from bio, A's always bond with T's, G's and C's always partner up together across the middle of the ladder. That's what makes the rungs. So if you look at the strand now, remember we said it has directionality to it. And a DNA strand, because it has two strands, if you look at how those strands go together, my sugars, remember we're naming this for our sugar in our, the carbons in our sugar ring, the 3' carbon, the number 3 carbon sticks out this end on this strand, and my number 5 carbon here is the end carbon on this side. If you look at how it goes together to make the ladder, the side, the matching strand on the other side, it runs in opposite directions. So here's my number 5 carbon here, and here's my number 3 carbon here. They run opposite directions, anti-parallel. So the 3 carbon end of this molecule ends up together with the 5' end of the matching strand. They run in opposite directions. And that's important when we talk about replication and copying DNA. We're going to get to more about that in our DNA chapter. So if you look at how those hydrogen bonds hold the two sides of the ladder together, make the rungs. Thymine and adenine, remember, always partner up, and they hook together with two hydrogen bonds. Remember, this is not a covalent bond. This is between molecules. Hydrogen bonds, there are two hydrogen bonds that hold those two together. And the C's and the G's, there are actually 3 hydrogen bonds that hold those two nitrogen bases together across the ladder. Remember, this is not a covalent bond. It's a weaker hydrogen bond. And we say they're complementary. So look at the connection. DNA that has more C's and G's in it actually have a little stronger connection across the middle than ones that have more A's and T's, because they have that extra hydrogen bond holding those C's and G's together. The structure of DNA was figured out by these two guys. We talked about this in bio, Watson and Crick, and they received the Nobel Prize for figuring out the structure. It was a big competition at the time. There were a lot of people in on trying to figure out what what was this structure? How did DNA go together? They kind of knew the components, but they weren't really sure the physical shape of it. And Rosalind Franklin was working in a lab, and Maurice Wilkins was in charge of the lab. She also was an important contributor to figuring out this structure. Linus Pauling also in America was working on the structure and trying to figure out. We're going to talk more about him later. He's got a pretty interesting scientific career. But he was also working on the structure of DNA at the same time. It was kind of a race between labs to see who was going to figure it out first. And Maurice Wilkins actually shared Rosalind Franklin's X-ray pictures with Watson and Crick, and it was pretty vital in them being able to figure out what was going on inside this molecule. He did that without her knowledge. Later on, when the Nobel was awarded, both Maurice Wilkins, along with Watson and Crick, received the Nobel for figuring out the structure of DNA. And even though Rosalind Franklin was a major contributor, she was not in on receiving the Nobel Prize, because by the time that was awarded, she had passed away from cancer. And they don't, probably from the X-ray dosages that she was receiving doing her research. But they don't award Nobel Prizes to people who are deceased. So she was not in on the prize, even though she was a major, her work was a major contribution to allowing these guys to figure out the actual structure. So the structure of DNA, we know, is a double helix. If you look at RNA, it's a single-stranded molecule. It's got ribose in its sugar spot. It's got A's and C's and G's. And remember, no thymine, no T's. It's got uracil for its nitrogen base. And this is a single-stranded molecule. Still makes the phosphate sugar backbone, and the nitrogen bases stick out the sides. But it is a single-stranded molecule. It doesn't have a matching anti-parallel strand. And once it gets made, though, it does form into some interesting 3D shapes that give it some different functions in the cell. Remember, that shape-function, structure-function relationship is so important in so many places. We talked about these kinds of RNAs when we did, when you were in biology. We're not going to go into details here, but I think you could dust off your brain, go back to the bio and review that a little bit if it's a little fuzzy. We are going to talk more about protein synthesis and how RNA helps that in a coming chapter later on, where we will do a whole chapter about DNA and the connection between transferring that info from the DNA out to the cell to make proteins. A new kind of RNA that we're going to talk about this year that we didn't talk about in bio is SI, small interfering RNAs, and they're very important in, we're finding out more and more about how proteins are regulated, and we're going to talk more about that when we get to our protein DNA chapter. So kind of just an overview. Remember, DNA is the genetic code molecule. That's a nucleic acid. And the connection between DNA and out to the cell are those RNA molecules. It's carried as a message out through the nuclear pores to the ribosomes out in the cell, where we use that information, the RNA information, to make a protein, a polypeptide chain that's going to fold into a protein and have a function in a cell. That's that central dogma of biology. DNA transfers information to RNA, transfers information to proteins, and that's how DNA expresses the genes, is making a protein that's going to do something in the cell. Another important kind of tied in with this idea of nucleic acids is we use some of those nucleotide subunits just by themselves or part of another molecule. One of the important ones we learned about in bio was ATP, adenosine triphosphate. It's got ribose in the sugar spot. It's got A for its nitrogen base, adenine. And so we actually could use this little subunit to build an RNA molecule. But if I don't put it into a chain and I keep it as a single molecule and add phosphate groups here, have these extra phosphates. Now, 3 phosphates, that's the TP, triphosphate. We talked about this molecule and why it's important for energy transfer and storage. Making the bond, sticking on those P's stores energy. Mitochondria are burning glucose and storing the energy as ATP. And then breaking that bond releases energy. It's our connection. It's a way we transfer energy in cells. It provides energy for chemical reactions to happen. And it's that hydrolysis, dehydration, synthesis to stick on the phosphates, make the bond, and then hydrolysis, add the water back to break the bond and remove that phosphate group, releasing energy for our cells. We're going to talk more about that in a lot of other chapters the rest of this year. Another place that nucleotides are used instead of building DNA or RNA is they are also energy storage molecules and transfer. And we talked about some of those enzyme helpers, electron transporters, NAD, NADP+, the FAD coenzyme A in respiration. All of those kind of storage, transfer, energy, electron carrier molecules have nucleotides as part of their structure. It's a component that goes into making the structure of that electron carrier. And so we're going to talk more about those when we get to our metabolism chapter, but just a reminder that those also have a nucleotide component that's part of their shape and helps them do their job. That's the end of our PowerPoint. Be ready to answer questions and know this information in class tomorrow and be able to use it in some activities.